So thank you for a wonderful, wonderful introduction, and it is a pleasure to be here, meeting old friends and making new friends, and generally interacting with one of the best groups in Europe, not only in terms of general science, but also in terms of understanding epilepsy and epileptology. So yes, the title of my talk today is Novel Mechanisms of Stress-Induced Disintegration of Dendritic Spines, Live Imaging and Cognitive Consequences. By being from California, the real title of my talk is Live, What Stress Does to Your Dendritic Spines. And the cast of characters that you see there are the people in my lab, and I'll show mainly today the work of Yunkai Chen, Courtney, Kristen Brunson. It's a very cosmopolitan and international lab, people from Israel, Hungary, Germany, what have you, even a few Americans. So let me start off with a punchline. What does stress do to your dendritic spines in your hippocampus? And here's the answer. I hope this shows with a funny lighting. So severe stress, and I'll show you what kind of severe psychological stress, over several hours destroys some of your dendritic spines. And what you see here is a hippocampal formation from a 3-month-old, a fully mature mouse that has been engineered to express YFP under the Tie1 promoter. So what you see here is a section taken from this mouse. You see the pyramidal cell layer, stratum lucidum, and here is the apical dendritic branching, radiatum, proximal radiatum, distal radiatum, and SLM. And this has been taken and this is just the orientation of the neuron, so you can see basal and the lamina that are associated with that, which I'm sure we're familiar with. This mouse has had the poor fortune of being restrained in a tube. With several other mice, it was put into a box that was shaken on a lab shaker for 5 hours. And the worst part of it is we played rap music to that mouse, which immediately got their stress hormones sky high. Also happened to do that to the investigators also. But anyway, after 5 hours of that, the mouse was sacrificed, and we asked the question, what happens to the integrity of hippocampal neurons? And as you can see here, these are the beautiful spines. This is confocal microscopy, and you can see dendritic spines. We are focusing here on proximal stratum radiatum. That is the area where commissural and associational fibers hook up to the hippocampus. This is the source of information that comes into the hippocampal neurons from specific parts of the brain, and also, of course, from the other side of the hippocampus.
So,
and as you can see,
was this East Coast or West Coast rap music? Because there's a difference.
I know so little about it. I get so stressed that I can't answer the question. It wasn't classical for sure. It wasn't what you played for me today. So this is a stress-free mouse, and this is spine density, of course, looked at blindly and so on and so forth after stress. So this is what it does. Now let's go to the beginning. Why are we interested in looking at the effect of stress on hippocampal structure and function? As you well know, the function of our neurons, whether you look at it in electrophysiology or in cognitive structure, is regulated mainly by a genetic program, by genetic makeup. But it is modulated by signals from the environment, by experience, by neuronal activity. You can call this elephant in any name you want. And this is the process of neuroplasticity. Now, if you want to think about signals or inputs that will have a very significant importance, that are really, you expect the brain to be able to respond to them, it is stress. Stress has a very strong evolutionary advantage for plasticity. And let me give you an example. You have, for example, 2 little monkeys sitting up there in a tree, or sitting below a tree, and they hear the roar of the saber-toothed tiger. And one of the 2 little monkeys end up being breakfast. If the second monkey, the one that survived, can remember the horrible roar of the saber-toothed tiger, can teach its neurons to act differently, the synapses that were involved can get strengthened. Plasticity of the synapses can evolve. Clearly, this monkey has a better chance of transmitting his or her genes to the next generation. So stress and stress-evoked plasticity can be a very advantageous process. In fact, the classical thing about stress is that it evokes bidirectional effects, classical for neuroplasticity. And stress has been shown by a large number of groups to influence synaptic plasticity, again, in a bidirectional manner. Acute or mild stress improves LTP, primes LTP. Studies from Germany and other labs have been very instrumental in showing that. And also, some of these mechanisms involve CRH. In contrast, chronic or severe stress causes abnormal LTP, problems with learning and memory. During adulthood, the effect of chronic stress even eventually disappear. However, we and others have shown that when stress occurs early in life, these effects can be permanent. And the effects consist of several elements. In addition to hippocampal synaptic plasticity, they seem to also alter the response to further stress. And depending on the type of experience early on in life, this can be either a good or, if you will, a bad effect. So let me familiarize with you with what we know already about the effect of early life stress on the hippocampus. Again, as I mentioned, it's bidirectional. And these are just some of the studies that I'm citing here. Some of them are in a human, where you see correlations or associations, and some of them are animal studies where causality has been shown. So again, mild stress, typically evoked by challenging stimuli, enrich and enhance synaptic plasticity, both acutely and long-term. In contrast, severe or chronic early life stress, in human it's been usually either loss of parents or neglect and abuse, and in animal models there have been a variety, and I'll show you one, has been shown to not only cause acute changes, but also reduce hippocampal volume in humans and primates long-term, and impair hippocampal function, sometimes starting later in life. Some of the best human studies from Finland and others suggest that there is actually a progressive decline. And that may very well be related to early dementia, as some people claim. The pseudo-dementia seen in depression, where stress is ongoing, and it's been invoked in potentially other correlations with other human diseases. So this is all nice and dandy. It's an important problem. We definitely need to study it. But talking about vague constructs such as stress and cognition and so on, how do we get to the nitty-gritty? How do we study effects of early life stress on the hippocampus? Which are the molecules that are involved? What mechanisms are involved? Which neurons are involved? And how are they involved? So we started off with a very simple-minded hypothesis, namely, the same molecules and mechanisms that we know are involved in generating the stress response may also be the ones that contribute to the long-term plasticity induced by stress. So what are these? Forgive me, but this is a neuroendocrinology 101. So the stress neuroendocrine system, when you and I perceive stress, the signal converges on the stress motor neuron, if you will, a CRH expressing, corticotropin releasing hormone expressing neuron in the hypothalamus, in the paraventricular nucleus. There is a rapid release of this neuropeptide that acts peripherally on the pituitary through CRF type 1 receptors that causes release of ACTH, and that releases glucocorticoids, corticosterone in a rat, cortisol in a human. These cross the blood-brain barrier, go back to the hippocampus, and function here. But in addition to these well-known effects of physical stress, most of the stresses that we deal with are not physical. They are high-order or psychological stresses. Either mild ones, like standing here in front of a group of my most eminent colleagues and trying to give a coherent talk, or more severe ones dealing with the boss showing up to work, financial issues, health issues. These do not go only to the hypothalamus and the peripheral structures, but convey through very well-established pathways to amygdala and to the hippocampus. And the effect on hippocampus, when you think about it, are not logical to be global, part of an overall brain response. It makes a lot more sense that there may be plasticity in the hippocampus that is invoked locally by local mediators that can be released locally, act locally, and synchronize or improve synaptic function in certain populations of well-defined groups that better define in both times and space. So one thing that we consider is that stress may release local mediators that allow rapid and fine-tuning of neuronal communication. That will be helpful in a mild event, both for the little monkey and after severe stress, may very well be responsible for the effects that I just showed you earlier in the talk. In order to address any of these questions, we can't do it in humans. There are wonderful correlative studies, again, many of them from Germany, from München, from Florian Holzber and others, but we need an animal model where we can look at causality. So, again, some things you can only do in the animal model. So we went looking for an animal model, and that was not as simple as it could. There are many animal models of early life stress. Most of them involve taking the mother away, which is problematic. The pups starve. In addition, it doesn't really model anything in a human. So we came up with a model of chronic early life stress, and I'll very briefly go over that. This takes advantage of the fact that the pup will be stressed when the mother is stressed. We stress the mother and create a very abnormal maternal behavior by, if you will, causing simulated poverty. We take most of the nesting away from the cage, so the mother cannot create a nice nest. She only has one paper towel to do that. Gets the mother very stressed, very fragmented, and this fragmentation is transmitted to the pups. We do that during one critical week between postnatal day 2 and 9, and for the sake of time, I will not elaborate why, which from the state of the hippocampus, hippocampal development is equivalent to early prenatal and early postnatal life in the human. So what happens? Yep, it is a model of early life stress. If you look immediately at the end of this 1 week, there is a marked elevation of plasma corticosterone and also an increase in adrenal weight, suggesting that the plasma corticosterone of the pups indeed comes from their own adrenal. And all this goes away. By the time they are 4 months old or middle age, as I'm showing you here, they look like perfectly respectful citizens. No evidence of stress, behavioral or any other. Their behavior is generally normal. They don't have increased anxiety, and that we have no evidence that something ever happened to them until we start looking at the hippocampus. So if you start interrogating the hippocampus for function, you see some pretty fundamental changes. Hopefully, most of you are familiar with the Moorish water maze test. It's essentially a test that uses spatial cues around the room to ask whether an animal can remember these cues to find a hidden platform. A rat is thrown into a pool. A rat will swim very well, but they don't like it very much, even in California when the water is pretty warm. So they will go perch on the platform, and you can test them, train them to do that, and on a third day, which in our case is a testing day, you can see how fast they find the platform. And you can see that at 4 months of age, there's very little, in fact, no difference between animals that have had early life stress for that 1 week or did not. But as they age, you see a pretty substantial divergence with evidence of both short and long-term memory deficits. There is a problem with this test. Although it is a very good test for spatial memory, it does have some adverse effects. Rats may not like being in the water, and maybe there's some subtle abnormality of their ability to cope with stress. Therefore, we chose another functional test that interrogates the hippocampus that is completely free of any adverse effects. This is known as the object recognition, and it takes advantage of the fact that rats are pretty curious. So on the first day, you bring a rat to the cage, and you present them with 2 objects, and they explore them. Okay? And this is true for both the control and the rats that have been subjected to early life stress. We then take them back to the animal facility, order room service, and bring them back the next day. And the next day, the normal rats, the ones that have not been subjected to early life stress, are bored with the object they've already seen the day before, but spend most of the time exploring the novel object. That gives us an indication that they can distinguish between them. In fact, they remember the object from the day before. The animals that have been exposed to early life stress do not have any preference, suggesting that they do not remember the object from the day before. In this particular graph, it's not entirely clear, but when we looked at total exploration time, it was no different. So in 2 different tests of hippocampal function, these rats have dysfunction. But what is the basis of this dysfunction? Well, learning and memory have typically been associated with long-term potentiation, and there's a large body of scientific work connecting them directly. And therefore, we collaborated with Gary Lynch's group to look at long-term potentiation in these animals. And we looked specifically at long-term potentiation in the commissural associational fibers, where, if you recall, I showed you reduction of spines after stress in the adult. And what you have here is long-term potentiation in 4-month-old rats, comparing controls and those subjected to early life stress. And as you can see, after this is high-frequency stimulation, you have a nice potentiation with increased both amplitude and slope of synaptic responses. However, when you look at these animals when they're 10 to 12 months of age, essentially they have no LTP. There is abrogation of potentiation of the synapses in these animals, concurrent and perhaps explaining the cognitive deficits. Why? Why is there no LTP, which then leads to or associated with significant cognitive dysfunction? We started to look at these neurons, and this is what we saw. If you fill and look at neurons, both in CA3 and this particular one is in CA1, from animals that have had early life stress and are now mentally deficient, if you will, and also have abnormal LTP, you see this paucity, this impoverishment of the dendritic tree again, in the same region that we started off with, in proximal stratum radiatum, where the commissural associational fibers hook up. So there is overall reduction of total dendritic length, which is explained primarily by reduction in complexity and total dendritic length in the apical dendrites. So what I've shown you up to now is phenomenology, and pretty intriguing and probably important phenomenology. Early life stress leads to impaired hippocampus-dependent learning and memory, loss of synaptic plasticity, and dendritic atrophy. And this is just 1 week of early life stress. Clearly, we've programmed a system here very profoundly. How the heck did we get there? What is the mess due to? So the working hypothesis that we have, again, relates to the fact that early life stress likely involves an excessive, this is chronic severe stress, involves excessive release of local mediators, and we propose to study specifically corticotropin-releasing hormone, and I'll show you why when. The reason we chose to focus on CRH, or corticotropin-releasing hormone, rather than on universal stress hormones, in addition to the fact that we found a local effect, there's no evidence of dysfunction in amygdala or other cortical function in these rats. So one of the reasons is that we looked for a local mediator. The other is when you look at the distribution of the receptor for glucocorticoid receptors, it's essentially absent in CA3. These are different manipulations of animals that we did, but basically there is very little glucocorticoid receptor in neurons in pyramidale and CA3, where most of the effects or major effects were found after early life stress. So it's difficult to conceive an effect of a hormone if the receptor is missing. In addition, we've found that there are a large number of CRH-containing neurons in the hippocampus. And you can see here, simply immunocytochemistry, of basket cells that contain CRH. Some of them are also chandelier cells. And you can see the beautiful network of axonal endings, the axonal terminals, that create a network of synapses on the pyramidal cells. And these are quite abundant. And Yung-Kai Chen in the lab used stereology to quantify them and found that there is a peak at about P18, but generally throughout life, there are thousands of CRH-expressing interneurons localized very strategically within the hippocampus, where they're, if you will, positioned to control information flow in pyramidal cells, or at least influence how they function. And the question is, what will happen if you have release of excessive CRH that would interact with receptors on pyramidal cells? But wait, are there receptors there? Yeah. So again, these are immunocytochemistries going all the way to electron microscopy. We looked at mRNA and protein and presence of the receptor. And this is electron microscopy done by Michael Frotscher in collaboration with us, where he finds the CRH receptor, shown here by immunogold, on dendritic spines of CA3 in this case, but also CA1 pyramidal cell. And they're located strategically, smack in the middle of the spine. In fact, if you look carefully, it is in the middle of the postsynaptic density, which is somewhat unusual to G protein-coupled receptors or metabotropic receptor. They usually stay outside of the Ph.D. and kind of be respectful. This receptor sits in the middle of the postsynaptic density. In addition, we also found that the machinery for CRH signaling is not only there, but stress releases CRH into the interstellar space. As you can see here, the red is CRH and the green is simply NON. We also found it associated with intercellular matrix. You can see that not only does stress release the peptide, but the release of the peptide is necessary for stress to activate, to get the signal across the pyramidal cells. What you see here, very briefly, is in a stress-free animal, we looked at phosphorylated crab as a universal marker of neuronal activation. It typically shows up in less than 5 minutes. And if you stain stress-free adult rats here, or developing, it's the same for both, for P-crab, you see very little activation of pyramidal cells. There is a signal here in the dentate gyrus, which is specific to a particular state of development of the dentate gyrus cell. So I would say this is a P18 or P20 type neuron. If you double label for the receptor for CRH, CRF type 1, this is particularly the receptor type that is present in the hippocampus, and P-crab, you see very little black P-crab in CRH receptor expressing neurons. After stress, you see that CA3 pyramidal cells are activated as measured by the very rapid turning on of the transcription factor CRAB. You can also see CREB in CRH-expressing neurons in high MAG. If you provoke the same stress, but into the brain directly, inject a selective receptor blocker for the CRF receptor type 1, the peripheral stress response is still there. You measure glucocorticoids, but you do not have activation of CA3 pyramidal cells, particularly those that express the receptor. So the machinery is there, and indeed, CRH is released during stress. Then what? Well, if you take pyramidal cells and expose them to CRH in vitro, and you can do that either in single neurons, or we chose to do that in organotypic cultures. Again, we made organotypic hippocampal cultures from mice that express YFP, so we can see the neurons directly. You can replicate what we saw in vivo with stress also by administering CRH in vitro. Again, you have a reduction of dendritic branching and chronic exposure of 100 nanomolar of CRH and even 10 nanomolar, although the effects are smaller, which are pharmacological but not horrendously so, causes dendritic retraction or atrophy. As expected, the opposite is also true. If you take mice that are lacking the receptor through which CRH may cause dendritic atrophy, they have exuberant dendritic trees, as you can see here from a wild type compared to a null receptor null mouse. The dendritic trees are much more complex, particularly in the area that we'll focus on in a minute, because this is where I'll show you the spines. Again, we're now zooming in down, trying to understand initially from a morphological standpoint, what is the mechanism by which this dendritic atrophy, which is found after early life stress, is caused also by CRH. So this dendritic atrophy is associated with spine loss. Again, chronically, in chronic cultures, after 1 week or 2 weeks of exposure to CRH, you see dendritic atrophy. And in the particular area of the dendrite here, you see that there is a reduction in the number of spines. Again, it's fairly selective to the area where in vivo stress also provokes dendritic and spine loss. Similarly, as expected, the knockout mice have a lot of spines in that same region. So again, you have the low magnification view of the dendrite, and in high magnification, you can see, first of all, many, many spines. Many of them, and I hope this shows, are really thin and look filopodia-like. Some are very abnormal with split heads and huge mushroom-like heads, very, very abnormal. And again, the density in the knockout is exactly the opposite of what you see when you apply the peptide. So we then ask the question, okay, so we think we may have a story that is coming together here. Stress may cause release of CRH, which will cause spines to go bad, perhaps, which eventually, if chronically sustained, will cause dendritic atrophy. If so, we should be able to see that. And we won't really believe the story if we can't directly visualize it. So Yukai in a lab developed a system where he could take an organotypic hippocampal slice culture and put it under the 2-photon and image it for hours in time-lapse microscopy. We've also verified phototoxicity, and I won't bore you with the details. We've excluded bleaching and phototoxicity in the confocal, in the 2-photon, and found very limited amount of it in the confocal. And what you have here is a steady-state, beautiful system. So if you image a hippocampal neuron for 2 hours and look at spines that form and spines that retract, you have the most beautiful life in balance, if you will, life and brain in balance. What you see here are the yellow arrowheads are spines that disappear, and they can do that in 5 minutes. So you have the spine here, it's absent here or very faint. Same here, the process of disappearance. And the spine will never show up again, otherwise it doesn't get a yellow arrowhead. In contrast, you have spines that form. And you can quantify the rate of spine formation and the rate of spine disappearance, or retraction, take your pick. And over the 2 hours of the imaging, you have a very steady rate. By the way, look at the overall spine density. It's actually fairly high. These are very healthy neurons. So you have a steady-state system where the rate of spine formation and retraction is about the same. Now we can ask the question, what happens if we superfuse CRH onto the system? Okay, and again, what you see when we do that, and I'll show you again the higher magnification, is you have no change whatsoever in the rate of the formation of new spines, but you have a dramatic reduction in spine retraction. So you have a net reversible spine loss. And let me show you that in more detail. So again, we have the same hippocampal pyramidal dendrite from area CA3 exposed to nanomolar amounts of CRH. And you can see that between 0 minutes and 5 minutes, there are a lot of yellow arrowheads. There is a massive reduction, massive increase in spine retraction, and no change in the rate of spine formation. And therefore, when you look at net spine number, there is a reduction. After 30 minutes, we take the CRH off and wash, and you see a marked increase in spine formation and a partial reversibility of this in the organotypic slice culture by the end of 30 minutes. Again, you can look at it and do other controls that come to mind. We can repeat the study in the presence of a specific antagonist of CRH, and we do not get the effects. In addition, if you grow the cultures in the presence of CRH antagonist, because there is endogenous CRH in the system, you get also a chronic dendritic increase. What we're showing here is, again, an acute experiment where you look selectively at formation and retraction, okay? And in the presence of antagonist alone, this is a selective CRFR1 antagonist, there is very little effect on either formation or retraction. The CRH alone causes massive increase in retraction, and the effects go away when you incubate the slice with CRH together with the antagonist. So this leads to some pretty obvious questions. I've shown you a lot of beautiful morphology, but how does this happen? How does a peptide that you probably never heard of before cause complete elimination of dendritic spines, which I'm sure you've heard of before, in 5 minutes? The idea that this is a mechanism that's unique and not interacting with other mechanisms that control spine morphology and integrity at this time frame is unlikely. And of course, what are the functional consequences? Let me just show you a little bit about what we know about mechanism. So again, what we know now is we have CRH that is released from interneurons that express it onto a receptor that's sitting smack in the middle of the Ph.D. People have shown that pKa and other kinases are involved in CRH actions through the receptors in general, but we do not yet know specifically in the spine which receptor is involved. Generally, people have shown that there are 3 Rho-type GTPases that interact and compete with one another to either stabilize or destabilize the spine by acting on actin, namely converting filamentous actin, which is a polymer, to a monomer. And this is regulated through some proteins, most notably cofilin, that sit on the actin and regulate whether it is amenable to degradation or not. In a phosphorylated form, picofilin, if you will, protects polymerized actin from degrading. And elimination of the phosphorylation allows this to act as an enzyme and causes actin degradation. This is the mechanism, by the way, that is believed to act during LTP and LTD when spine morphology can change as quickly as in 5 minutes. So it's an attractive mechanism for which CRH may interact with the process. We don't have a lot of information yet on what happens here upstream, but we do know what happens downstream. So after exposure to CRH, within 5 minutes, we see elimination of the inactive phosphorylated form of picofilin and conversion into the active actin-degrading form, if you will. So again, I don't know to what extent this projects, but you have here the red dots and also red dots on top of yellow spines, which means picofilin in the spine head, okay, attached to the actin in the spine heads. After application of CRH, there is a reduction in phosphorylated picofilin and appearance of cofilin, not only in the overall slice, but also taking into consideration the fact that there are less spines after CRH, so that per spine you have a progressive reduction in picofilin. We think that this contributes or provides some evidence to the fact that the peptide and the receptor signaling act through actin degradation to cause very rapid retraction or elimination of dendritic spines. I don't have yet good answers about the acute cognitive effects, and we'll talk about that hopefully next time. The question, and we do have some information though, is the chronic effects, the chronic early life stress effects, and is CRH contribute to that? We had some early evidence that might be the case, because if we gave, instead of stress early in life, CRH to immature rats, we were able to recreate precisely the effect of early life stress. So I'm showing you here just in brief the Moorish water maze at 1-year-old rats. They were treated here not with stress, but with CRH. And as you can see, they have a difficulty finding the platform. Similarly, on the second day of the object recognition test, when vehicle administered adult rats that were given vehicle early in life clearly distinguished between the two objects, the animals that have gotten CRH early in life did not. So that was suggestive but not particularly helpful. We also know that the early life stress increases the level of CRH in the hippocampus progressively and permanently. So there is a lot more CRH in the hippocampus, particularly in area CA3 of animals after early life stress, which means that every time the animal is perturbed a little bit, a lot more peptide is going to be released, activate more of the receptors, and perhaps start the process of dendritic spine retraction, eventually dendritic atrophy, and cognitive dysfunction. Finally, we asked the question, if indeed CRH is involved in the mechanism by which early stress contributes to the dendritic atrophy and cognitive effects, then that would mean the blocking the receptor at that critical time, optimally after the stress, because in real life we cannot prevent it, that should eliminate all the consequences on dendritic integrity, spine integrity, and cognitive deficits. So what I'm showing you here is if you take rats after early life stress, again, you've seen this a number of times now, they have difficulty in a Moorish water maze. If you administer through a LZ pump and CRH receptor blockers after the period of stress, between postnatal day 10 and 17, you essentially recapitulate, and somehow this does not show. All right. Well, there is a light blue line here that is not showing neither here nor here, and shows that the Moorish water maze is returned to control levels. And unfortunately, this is also not visible here. So with this in mind, I will go directly to where the money is and ask, what happened to the LTP? And hopefully that one will show up. Yeah, it does. Okay. So not only were we able to prevent or eliminate the cognitive effects in hippocampal-dependent learning and memory of early life stress with the CRH antagonist, but we also prevented the effect of early life stress on long-term potentiation. And just to recapitulate, in control animals at 1 year of age, middle-aged rat, you have long-term potentiation you see here in the white dots. After early life stress, and I've shown you that before, it is completely abrogated. And in animals that after the early life stress, you administered CRH receptor antagonist, you return the LTP to its normal levels.
Tallie, sorry, when was the blocker administered and when were the animals sacrificed? How long after the stress?
Okay, so the stress was between postnatal day 2 and 9. The antagonist was given at the time when we felt the hippocampus is still vulnerable, and we'll talk about that in a minute, starting on postnatal day 10 to about 17, and the animals were sacrificed at 1 year. Right, so, what do we think is going on? Well, we think that early stress or administration of CRH, they do the same thing, increase hippocampal CRH levels. There is excess of hippocampal CRH release both early on, where we believe it eliminates dendritic spines on proximal radiatum pyramidal cells and interferes with the ability of associational fibers to form normal connections. Unlike the mossy fibers that sprout and look for connections throughout life, the associational fibers cannot do that after about postnatal day 12 to 13. This is very old work by Chris Gall. So that you never have normal hippocampal connectivity. Indeed, when you look carefully at the electrophysiology of the CA3 pyramidal cells, they're partially denervated and hyperexcitable. Okay? And for the lack of time, I didn't show all of these details. In addition, there is an elevation of CRH levels throughout life, which keeps on acting on receptors in the remaining dendritic spines. And this, again, over time, causes spine loss, synaptic dysfunction, and the dendritic atrophy and loss of LTP. Eventually, this even transmits from CA3 to CA1, as expected if you have an abnormal neurotransmission in the Schaefer collaterals to CA1, and eventually you get cognitive impairment. So we have evidence, again, to suggest that early life stress creates this, that hippocampal CRH levels are increased, that CRH is released in the hippocampus, that too much CRH causes spine loss, and that abnormal synaptic dysfunction results. And blocking this abnormal cascade by preventing the long-acting increase in CRH levels in the hippocampus, essentially abrogates the whole cascade. Some elements of this cascade still require further experiments. Now, of course, this particular type of experimentation is highly relevant to human health. UNICEF has finally come up and reported that over 50% of the world's children grow up under chronic early life stress, be that neglect and abuse, whole populations starving, massive migration, war, famine, AIDS, you name it. And we cannot prevent this, at least not any way that I can see. But if there is a way to understand the effect of stress on the hippocampus and to think about post hoc intervention to prevent the cognitive effects, I think this is a very important thing to answer. And we believe that the CRH receptor may very well form a good drug target. And this may be very important to the rest of the world, but I bet you that that's not what is going on in your own brain. Because the last and the most important question to the audience sitting here in front of me is probably, does CRH contribute to that original thing you showed us in the first slide, namely to the effect of stress on my hippocampus, on my dendritic spine? And can this be reversed? So let me now show you the whole picture, which I did not show in the first receptor, in the first slide. So you've seen this already. This is the spine loss after the 5 hours of stress that I've shown you before. This is the bad news. The good news is if we take the same mice that have heard the rap music being restrained and being subjected to noise and jostling, and pre-treat them with CRH receptor antagonists, not peripherally, but directly into the brain. So they still have the peripheral stress response. If you measure corticosteroid or glucocorticoids, they're just as stressed as the other guys. But they have had a local blockade of the CRH receptors in the hippocampus. You eliminate the ability of stress to mess up the dendritic spines. I don't have any shares in any of the companies that are making CRH receptor antagonists. I'm letting you know now. So again, that suggests that there may very well be a mechanism to consider eliminating the effect of semi-chronic or hour-long severe stress in our own adult life. So to summarize the story the way we know it now, first of all, stress impacts hippocampal neuronal function in a bidirectional way, and today I focused on the effects of severe, excessive, chronic stress both early in life and during the adult. Indeed, acute severe stress in the adult results in spine loss, and this is at least in proximal radiatum through CRH-CRF1 signaling. In chronic stress, this causes dendritic atrophy and cognitive deficits that we haven't shown you, and these are generally reversible. Eventually, even chronic stress in the adult, you take the stray away and there's recovery. However, when chronic stress occurs early in life, it sets in motion a chain of events that is irreversible. Essentially, there is permanent and progressive loss of the dendritic integrity, probably spine loss, although we don't know for sure, and progressive cognitive deficits and progressive loss of LTP. Again, we believe that CRH, based on what I've shown you now, is involved in these effects. And this suggests together that CRH receptor signaling provides a target for prevention or reversal of the effect of stress on the hippocampus of these rats and also on yours. For the fine anatomy of the hippocampus, it's a problem. It's about 100 to 150 micron. How does a peptide, if it is indeed released here, and there's clearly some peptides that seem to be released here, and I think there's a fair amount of electrophysiology suggesting that it does. But how does a peptide travel, this is pyramidally, through lucidum to get to where the receptor is? And the answer is I don't have a clue. There is a lot of nice, what Americans say, hand-waving, because peptides are supposed to be transmitted by volume transmission. And in the whole peptide literature, this is sort of taken for granted. We do know, by the way, that there's no degrading enzymes for CRH. There's actually a binding protein that probably stabilizes it. So as I showed you earlier, you can see stress-induced release of the peptide just sits there. And you can do immunocytochemistry on it. And presumably, it might be moved by volume transmission. I like this about as much as you do, because I would like to really see perhaps dendritic release of the peptide somewhere here, okay, where the dendrites of the same, as you can see, the dendrites of these interneurons actually reach pretty far into radiatum. And I don't know if this is the case, but Susanna and I have talked about this a little bit earlier today, and hopefully we'll find out if this occurs also. So the two obvious possibilities are either released from axon terminals in a canonical, classical way, and transmission by volume transmission, or indeed dendritic release, which has been described like in the hypothalamus or elsewhere. And I think actually in the hippocampus too for dynorphin, as a work of a guy called Shafkin. And it'd be interesting to know if this is the case, and we're looking at that right now. In fact, Michael Frocher is doing the great question. So the first one we have thought of and have not done yet, because you need to go really into very low nanomolar concentrations, and this is something we're planning to do but have not done yet. I think the answer will be that we probably will find a biphasic effect. The U-shape curve of hormones in general has been described, and I think your hypothesis is wonderful. That's something we need to look at. Again, the clinical effects are clearly there. The peptide, pre-primes LTP, the work of Blank and Spies, and so on, but we haven't done it. The other question, remarkably, very few people have looked at. We're beginning to look at that right now, but we haven't done it systematically. And first of all, it depends. Okay, two things. One is no one knows really well. In general, they're a little bit stupid, which means that too many synapses are probably not very good for you. And if you look at the spine, they have really the word elongated and philopodia-like or very bizarre. So the mice are not smart. I don't personally know how stupid they are because it has not been looked at very well. LTP has subtle changes. Zittelganger, I'm probably not pronouncing the word right, from the Holzberg group, has tried to do some physiology on the mice, and it was not straightforward. Genetic makeup may also play a little bit of a role here because the Americans have one genotype in Germany, and what we have is a different genotype. There's no advantage to having too many spines and too many synapses, but the details of exactly how impaired they are, I'm not sure. In the model of early life stress, where we see spine loss and dendritic atrophy, we do not see neuronal loss. However, when we mimic that or try to mimic that by giving CRH early in life, we may have overshot. And in that effect, we saw the dendritic atrophy and also loss of a few neurons in CA3. And so the answer was yes. So I think it's a question of degree. Early life stress, in all the animals that we've looked at, we see spine loss, dendritic atrophy, but no neuronal loss. If you perhaps have pushed it to maybe even more severe stress, which I wouldn't even know how to do, then eventually you might get neuronal loss as well. There are many different ways in which stress signals, if you will, reach the hippocampus. Lupus is a very good possibility. Raphe will also, I think, synapse directly on the radiatum. This is where the associational fibers come in. So I think it may be very well one of those. We are now beginning to look at whether serotonin receptors have anything to play with the system, but I don't really have results yet. It's very nice. So these is a 12-month-old in situ hybridization. And we also found it with immunocytochemistry, and I'll tell you the results. I just found them last week, so I didn't have time to do that. If you look at the overall expression levels of CRH, this is with emulsion or direct in situ, in area CA3 in the hippocampus, there is a lot more CRH there. It does not seem to be ectopic, namely, there's still, when we did now immunocytochemistry with double label with GOD, all of the CRH expressing cells seem to be GABAergic, but we seem to see more of them. I don't know if it's more cells, we can certainly count more, or they're making more CRH so we can detect them. But once you have stress between postnatal day 2 and 9, you set point the system so that your hippocampus, particularly area CA3, will make more CRH throughout life, which means that, again, here is where we get into a little bit of a hypothesis, upon any ambient situation later in life. I mean, this rat gets brought into the lab, or I need to come stand here in front of Heinz Beck. I will have more CRH being released because there's more there. The idea is that the denervation of the CA3 pyramidal cells, or the partial denervation, is the trigger for increase in CRH expression in them. As you recall, what happens during the week after the stress is this is the time frame where, okay, how shall I do that? During the early life stress, we see loss of spines. These spines are crucial for normal innervation of the associational fibers because these are the place where the associational fibers anchor. If you cannot have this normal connectivity, this normal anchoring, then the associational fibers will never find the targets. Okay. And we have physiological evidence, electrophysiological evidence, that the CA3 pyramidal cells are denervated. I think, and this is up to here, it's fact, from now on, I think there may be a trigger for upregulation, persistent upregulation of CRH in interneurons around them, because these interneurons may very well also make more GABA. This is hand-waving. I don't know what the trigger is. I have a series of facts and don't have the whole story to connect them. But the fact that there is more peptide there, and significantly more, and it's actually more prominent in immunocytochemistry, we know that. So this is an interesting question, but let me kind of backtrack here for a minute. We see the upregulation of CRH expression only in area CA3. What you see here is in situ, and this is why I'm showing you the emulsion here. This is essentially dense nuclei, which is intrinsic in situ hybridization of mRNA level. This is not a real signal, and this is why we didn't quantify it. When you do it with emulsion or when you do it with immunocytochemistry, which is better because you see the actual peptide, there is nothing in dentate gyrus cells. They actually don't express CRH at all. So it is selective to the basket cells around the pyramidal cells in pyramidal cell layer. When we look at cortex, amygdala, I think these are the only regions that we looked at. We don't see any upregulation. It seems to be selective to the hippocampus. So therefore,
But you showed some immunohistochemistry, and there it looked that in the dentate gyrus it changed so that the inner leaflet is becoming very much strong.
Don't think so. I think you looked at PQAB. There was no CRH. Yeah. There's no CRH receptor in the dendrites. Let me bring that back. You're looking about this one, yeah. So this is PQAB. Yeah, this is PQAB. Yeah, this is PQAB. And actually, Roland Bender, when he was a lab, published on this. It's a very interesting phenomenon in which PQAB is transiently upregulated in granule cells at a given state of development. And Roland is very interested in granule cell development, so he tried to figure out what this does, and it's BDNF and stuff, but it's not relevant to us. Okay?
On the right side is the dentate gyrus.
This is all CA3. This is all CA3. This is all area CA3. So the question is, the CA3 pyramidal cells are denervated, and again, the electrophysiology in the Brunson 2005 paper. So we asked the question, do we have any evidence for that? Well, as you know, the two inputs, two afferent systems to the, or many more than two, but two that are relevant here, to the CA3 pyramidal cells. One is the mossy fibers coming in from the granule cells, and as you know, these can sprout, and sure enough, they sprout. Whereas the associational commissural do not sprout. We have yet to go back and see if we can localize the origin, which I think is going to be pretty much diffuse around the brain, to these associational commissural ones, and look whether they have any changes occur to them. They're much more difficult to look at. So we went for the low-hanging fruit first, if you will, and looked at the mossy fibers, and yes, they are sprouted. And again, I don't have the pictures, but it's in the PNAS paper, and actually also in the Generosci paper of 2005. That's really a good question. Thanks. As you know really well, Christian, human literature, particularly epidemiology, is problematic. I mean, again, statistics suggest to us that the Baltic Sea is 4 feet deep, so you cannot drown on it if you look at the statistics. You know in epilepsy that if you look at old children with febrile seizures, the huge epidemiological studies don't show any cognitive deficit in any epilepsy. So I think we need to take this with a grain of salt. Having said that now, there are really good human studies substantiating the fact that early life stress, particularly psychological stress, and what people had when they were born in Germany between those years may be a little different, and we'll get back to this in a minute, does affect the hippocampus. The classical and most prominent one that showed up recently is the Romanian orphan study that came out in Science at the end of 2007 or early 2008, where clearly there are cognitive deficits. When you look at populations where you can really distinguish under the same conditions, early life stress or not, for example, in Finland, or more recently in studies of early dementia by Wilson et al., that came out last year, you can see that early life stress, when you use poverty where the rest of the society is not poor, so the people know that they're poor, or neglect and abuse, do have cognitive deficits. And again, oftentimes they show up later in life. So I think the human data is much, much more difficult to tease out because you don't know if somebody is poor, perhaps because the genetic makeup is poor, and this is very, very difficult. The kind of stresses that occurred in Holland, again in those years of war, and in other situations where the whole population undergoes a stressor is different from what we're doing here. The thing that typifies neglect and abuse is its unpredictability and fragmentability. If we are in a situation that's not very good, but it's constant, we adapt to it. Whereas if things are unexpected, unpredicted, such as like the behavior of a cocaine-abusing mother, which is essentially what we model here, there is no way to adapt. Things come and hit you all the time. So with this in mind, I think that the model is relevant to what happens in a human. But you're right, it depends what kind of question you ask, what kind of stress you're dealing with. So we are referring to stress that's unpredictable and therefore non-adaptable in children, such as neglect, abuse, and some of the horrors of moving, losing a parent, and so on. So with this in mind, I think the model is relevant. How far can we apply to the human? I think your question is really, really good. A rat is not a human. A rat allows us, or a mouse, to do some things that we cannot do in a human, but clearly is highly limited. I think the possibility that the peptide is involved in stress is intriguing because one can test it therapeutically, and it may very well be effective in some but not in others, because again, in humans there are so many mechanisms. So I hope that answered one or two of the questions. The mild enrichment, the very mild stress of enrichment, we find this in animal models. It has been reported in humans, but again, it's difficult to control for that. In the same Romanian orphans, for example, when they gave them enriched environments, they claimed to see improved cognitive function. I think in monkeys, in primates, it certainly is the case. But in humans, I think the caution of Christian Elger is well taken, and I should probably not go beyond that. So in primates, you get the same situation, where the mild stress is stimulating. Yeah.
Okay. Thank you very much, Tallie.